Hello and welcome to DNA Today. I'm your host, Kira Deneen. DNA Today informs you what's happening in the genetic world. The show is packed with news stories and lessons, all to keep you updated and educated on what's happening in the world of genetics. Again, I'm Kira Deneen. You're listening to DNA Today, and I've got quite a few things for you today. I am talking about I'm reviewing a UCONN presentation here from a assistant professor from the University of Cornell. And I'm also talking about a few news stories. I'm talking about highlights from Cleveland's Clinic Medical Innovation Summit. And there's summing up into quite a few innovations in cancer therapy, which obviously has huge genetic impacts. Also discussing the microbiome and a specific news story talking about that. And another news story I'm going to be having a little bit of discussion on is a longevity study and genes that have an impact on how long a person lives. And it's a specific research that had to do with people that were over 95 years old. So pretty cool study. And then wrap up the show. So that's what you have to look forward to. Let's jump right into it. So last week, I failed to know about these things before my show. So I tweeted out to everybody, so you can follow on Twitter at DNA Podcast, and you can get all of the updates on there of all the genetic-related things open to the public that you can attend here at UCONN. And I went to a presentation, as I mentioned, from Dr. Andrew Grimson from Cornell University, and he is an assistant professor of molecular biology and genetics. And his presentation was called The Messenger's Tale, Decoding the 3' UTR. And that might sound intimidating, but I'll break it down for you and kind of have an overview of what that means and what his research was and kind of what he concluded. But more information about his lab. The Grimson lab focuses on post-transcriptional gene regulation, and in particular identifying and the identity and function of animal microRNAs and other small RNAs. And I'll go on to explaining what this means. So the presentation in particular focused on the 3' UTR. So I'm going to break down what a 3' UTR is. UTR stands for untranslated region, meaning that these base pairs, again, base pairs are A, C, T, and G. These base pairs don't directly code for proteins. They're like the non-coding part. But it's not to say they don't have functions. They may not translate eventually to proteins, but they have other functions, regulatory functions. And the 3' portion of this term is referring to the 3' side of the DNA molecule. Now, this DNA molecules have two ends. They have a 5' and a 3' end. And the numbers are taken from the numbered carbon that is found on the very end of that side of the DNA. So the 5' end is the leader sequence, while the 3' end is the trailer sequence. And that's why we read DNA 5' to 3'. So that's kind of a background of what a 3' UTR is. So it's that 3' part, which is the trailer end, and UTR is for untranslated region, which means that it's not necessarily coding for a protein. It has other functions. So the Grimson lab research focuses on the 3' UTR, and they were surprised when they discovered their research supported this concept that the 3' UTRs had a regulatory impact. I mentioned this before, regulating which genes are expressed. So expression, that means what genes are being transcribed and then translated. And expressed, meaning that it's going to be turned into proteins and be seen. So to research 3' UTRs, Grimson Labs did a comparison of a specific 3' UTR called HMGA2. HMGA2 3' UTR. Now this comparison was between different species such as humans, mouse, and chickens. And in general, regulatory sequences are found to be similar among other species. And this concept of comparing genes between species is known as comparative genomics of 3' UTRs. And so comparative genomics you can be comparing really anything to different genomes of different species. And if the sequence being analyzed appears in all species studied, or a lot of them, it implies that there must be a function of that sequence. It's also worth noting that regulatory sequences are conserved in different cell lines, and a cell line is a group of cells that are reproduced from a single cell and have identical genetic makeup. So these regulatory sequences are conserved in these cell lines that if you're starting with, cell A and it reproduces into cell B and C and then, further and further, this regulatory sequence is conserved. And so here are the two big conclusions that Dr. Grimson highlighted. The first was that through this research, many regulatory sequences were identified. So these regulatory sequences I'm talking about, that the 3' UTRs were identified as regulatory sequences. And the second was overall regulation is a composite of many small effects. So regulation, it's not just like, oh, you have one thing and that's going to mean that this gene is going to be expressed or not expressed. It's unfortunately not that straightforward. So regulation is a lot of different things coming into one to see, is this going to be expressed or not? And obviously genes can differ, but speaking generally, small effects, a composite of any small effects, have that regulation. So if you want to know when these presentations are, again, you can check out Twitter at DNA Podcast. And I'll tweet out, as soon as I find out about it, I tweet it out. That's kind of my routine by now. And I put it in my calendar, and it's really great just to hear from these professors, and it's a free event. So it's fantastic that UCONN is able to bring in these kind of big names and just be able to experience, even if you don't really understand everything that's going on, it's great to really hear from them and hear their research and kind of just get used to the lingo they use and all of the jargon. And you do end up picking things up, especially if you're taking classes and then they mention something you're studying. I get really excited. I'm like, oh, we're studying UTRs, and this is really exciting. I can apply like the knowledge I'm learning to understand at least part of what he's saying. So that's it for the genetic roundup of UCONN. Now talking about the genetic roundup of the news in a bigger world of genetics. My first story I mentioned is highlights from Cleveland Clinical's Medical Innovation Summit. And there's an article on health, on US News, their health subsite, and it summarized 7 innovations in cancer therapy. And I'm just going to share a few of them because I don't want to spend a long time going through this. But I'm just going to share the ones that really had a huge genetic component to it. And the first one was personalized medicine. Personalized medicine is something that I've talked about quite a few times on the show and will even more so in the future. Michael Roizen is an internist who chairs the Wellness Institute at Cleveland Clinic, and he shared a quote I want to share with you. A lot of what we have learned is that the one-shotgun approach for all breast cancers or lung cancers doesn't work, and tumors have considerable genetic variability that indicates that certain treatments are appropriate. I've talked about this, end quote, by the way. I've talked about this concept of that if someone has cancer, it's not necessarily the same in everybody. Breast cancer is not one identical cancer in everybody. So different cancers have different components to it and different medicines that would work and different treatments that would work. So by identifying the differences between these cancers, personalized medicine is kind of a targeted medicine for the really specific type of cancer that you have. And ultimately, hopefully, we would get to this point where personalized medicine is more used. And we're seeing even in the news that like on TV, sometimes there's commercials for personalized medicine. So this is really becoming something in the mainstream. And genomic blood tests can determine which treatments would be most effective in fighting the cancer. And this spares patients unnecessary and toxic drugs. If we know, all right, we have a better indication that drug A is going to work, why try drug B and C? Let's try drug A first. Another quote I want to share with you that he said was, quote, "Genomics is just now beginning to reach its full potential," Roizen said. "I think that this is the year of the breakthrough of individual treatments for tumors." So again, just kind of what I was talking about, that we're really individualizing medicine, and that's what personalized medicine is all about. So I hope to really touch on this topic a lot, and if you have any questions or any specific things within personalized medicine you'd like me to talk about, you can email in at info@dnapodcast.com. So another innovation in cancer therapy that was mentioned at this summit was immunotherapy, and this is understanding tumors' genetic profile, and it can unveil how they evade our immune system. Now, Roizen mentioned that quote, "when they learned that we'll have a large ability to go past where we are right now." So already there's been headway in training the immune system to fight cancer. And in 2011, a drug emerged for the treatment of multiple melanoma, and a form of skin cancer that, I'll quote Roizen again, saying that was once a death sentence. So before, he's saying that this used to be, if you got multiple melanoma, it was kind of like, all right, unfortunately, we will not be able to treat this. But now, this drug has greatly extended the longevity of melanoma patients. And Roizen shared that, "I expect this is the start of it, that the class of drugs has huge potential to use." So genetics is rapidly expanding, especially within cancer, because cancer is so big and everybody knows someone that's affected by cancer. So if we can use these techniques to really focus on medicine and developing things that are more targeted, then we're going to find more success. Another innovation mentioned was selective genomic testing. And Brian Balwell shared, quote, "If we test large populations of patients, are we equipped to handle the information? And if you can't change one's risk, how much of a service or disservice are you giving to patients?" With the test, obviously. And Brian Balwell is the chair of Cleveland's Clinic Taysachs Cancer Institute. And he added that deciding how much testing is used is a clinical challenge. And if I've also touched upon this topic, that genetic testing, if you can test for something and you can prevent something from happening, in my opinion, I would personally do that. I would, if I had a family history of something and it was something that I could prevent or greatly treat, that my life would be much better, I would go for it. But more controversially, having a genetic test for something that you can't do anything about, well, is that a service or a disservice that you're giving to patients? And so this was talking about, selective genomic testing. Should we do this or shouldn't we? Should we be more selective in what we're testing, what we're telling patients? Because it can affect them mentally, that they're like, oh, I could get this. Like, I tested positive for this, or I'm going to get this, and there's nothing I can do about it, really. In certain ways, you can kind of prepare your life for where you're going. And if you have Huntington's, for instance, then you're going to rapidly decline at some point, that you can kind of prepare your family and all of that. But other than that, there's not much you can do. So this is definitely a very, very controversial topic that has a lot of layers to it. It's definitely not easy to, I'm not doing it justice in the debate, but that's just kind of skimming the surface there. And the last innovation I wanted to talk about was teaching cancer cells to behave. And we all have oncogenes in us. And an oncogene it promotes cell growth, but to the point where it's out of control, and that's a lot of times causes cancer. So Balwell says that we all have oncogenes in us, and only in some people do these cancer-causing genes reach their full potential as cancer cells. And while most treatments have focused on killing these cells, recent research has focused on changing the nature of cancer cells. And he poses the question, can you in fact take a cancer cell and turn it from being malignant into something that doesn't grow uncontrollably? And there is encouraging trial data on this approach, and it was presented last summer at the European Hematology Association conference. So being able to manipulate cancer cells is obviously a huge part of this, of preventing cancer cells from going out of control. And, with those proto-oncogenes is what's naturally in us that are, it's a good thing that it's promoting cell growth, but it can take a wrong turn and become an oncogene. So those were the top highlights there. Moving on to our next news story. Previously on the show, I have discussed the microbiome. Dr. Rachel O'Neill and I discussed it last week, and I've mentioned it quite a few times through the presentations I heard at the grand opening of Jackson Laboratories. And so I saw this news article, and I was like, I definitely have to include this. This is by medicalnewstoday.com. And just to jog your memory, and to quote Joshua Ledenberg, because he really puts this nicely, the microbiome is the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. So this is all the good beneficial bacteria that coexist within your own cells in your body, and we need this, like probiotics. Some people take probiotic pills, or you eat yogurt, and you're getting that good bacteria that really helps you. And new research through a twin study has found further evidence that weight is controlled by gut bacteria, which is part of the microbiome. So that's how the microbiome fits in. This gut bacteria is a small portion of the microbiome. And it reveals that genetics can shape what type of bacteria exists in the gut. And due to this type of bacteria, it can affect a person's weight. So how much of this bacteria they have has a direct correlation, according to the study, of how much a person weighs. Previous to this study, it was believed that a variation in gut microbes were due to external factors only, such as diet, environment, lifestyle, and health. But this study that I'm talking about is the first one to find results supporting that it's heritable. And heritable means that a trait is transmissible from parent to offspring. parent to child. That trait is being passed on. So this twin study was conducted by researchers from Cornell University in Ithaca, New York, and King's College London in the UK, and it was published in the journal Cell. So you people that actually can read these things and digest it, you can go check that out in the journal Cell. So how did these researchers come to this conclusion that the more of this specific gut bacteria you have, the more of a healthy weight you are? They started by sequencing the genes of microbes present in over 1,000 fecal samples. And these samples were taken from 416 pairs of twins who were part of the Twins UK data registry. And these twins were both identical and non-identical. They had 171 pairs that were identical and 245 that were non-identical. And the reason for utilizing twins and the two different types of twins is because of the amount of genetics they share. So this is something that comes up in a lot of studies, particularly twin studies, and this is the reason why they use them. So identical twins share 100% of their genetics, whereas non-identical twins share close to 50%. So if identical twins are considerably more similar than fraternal twins, which is found for most traits, it implies that genes play an important role in these traits. So you can see the relationship that if identical twins are having way more sharing a trait as opposed to non-identical, then you're like, okay, this definitely has a genetic component to it. So going along with this idea, identical twins had more types of gut bacteria similar to themselves compared to non-identical twins, so non-identical twins had different types between the two twins, and identical twins had more of a similar type of gut bacteria. So this indicates that the genetic influence of the gut bacteria, like I said, identical twins are having similar, and so because of that, they have more similar than the non-identical. This is going to have a genetic component. So a specific type of bacteria, Cristinellacea, was found to have the most genetic influence. And get this strain of gut bacteria was most common in people that have a low body weight. So not only was this strain of bacteria the most genetic influence, it also was the most common in people that have a low body weight, which suggests that increasing the amount of this bacteria strain in the gut could help reduce or even prevent obesity. And Professor Tim Spector, the head of the Department of the Twin Research and Genetic Epidemiology at King's College London, shared his insight on their findings. Quote, "Our findings show that specific groups of microbiomes live in our gut could be protective against obesity, and that their abundance is influenced by our genes. The human microbiome represents an exciting new target for dietary changes and treatments aimed at combating obesity." So according to the research team, the results may open the door to personalized probiotic treatments. Again, I'm talking about personalized medicine here. So this could open the door to personalized probiotic treatments and could reduce the risk of obesity and its related diseases. So I've had quite a few news stories talking about obesity and the genetic components to that, and this is just another example of that relationship. And specifically here, we're talking about gut bacteria and how it can affect weight and how much genetic component it has. Our next news story here is talking about longevity. So even among people who live well into their 90s, a new study finds that those with a particular gene variant may survive the longest. This variation is in the CETP gene. So the CETP is involved in cholesterol metabolism, and the longevity-linked variant raises blood levels of HDL cholesterol, and this is the good kind of cholesterol, and it promotes longer than normal HDL particles. So HDL stands for high-density lipoprotein, and it's one of the 5 major groups of lipoproteins. And again, this is the good kind of cholesterol. A lot of times when we hear cholesterol, like, you don't want a lot, but like this is actually one that promotes longer living particles, longer living high-density lipoprotein particles. And Dr. Sophia Millman shared quite a bit in this, so I'm going to be quoting her throughout this news story. She said, quote, "The new findings show that even when you look at people who already live beyond the age of 95, those with the favorable CETP variant survive longer." Again, the favorable CETP variant is producing, it raises the blood levels of HDL cholesterol, which is the good kind. So that's what this gene is doing. And forgot to mention that Dr. Sophia Millman is an assistant professor at the Albert Einstein College of Medicine in New York City. And so this study actually took place, and the group study for this research were centenarians around New York City, all of Ashkenazi Jewish descent. And researchers found that these extreme elderly people often carried the CETP variant and, again, have high levels of HDL. And researchers have been studying centenarians in and around New York City, and they found that they don't only live longer, they also live healthier. So it's not just that they're having more years in there, it's their quality of life is better. And Millman went on to share that these latest results are based on more than 400 people from the Einstein Project. So they have a huge base of people that they're really studying. So it's not just like, oh, they found a couple people and they're looking at it. Like, they really have a big group of people to study from. So it makes their results even more accurate. And they were typically 97 years old when they entered the study and were followed anywhere from 1 to 11 years. That's a direct quote from her. And it was a general trend that centenarians with the favorable CETP variant and relatively higher HDL levels survived longer. So again, I can't stress this enough that it's this variant is causing the HDL levels to increase, and then these humans are surviving longer. And these two factors are contributing to longevity. But obviously, this isn't the only thing that's contributing to it. It's not like, oh, you're 95, you must have this variant. There's a lot of things that are contributing to it, but this one is a very, obviously the research is pointing to it, and it's greatly explained. So that was an interesting study. I really like seeing that, especially I have pretty good genetics, I think, for myself for longevity. I have some family members that are pretty old, and it's impressive, and I'm interested in that just, from a personal standpoint. But we have a little bit of time, so I want to highlight a genetic disease. And this week I want to highlight Taysachs. So far I've been highlighting more well-known genetic diseases that are rare. And so Taysachs is known also as TSD. And this is a fatal disorder where a baby doesn't show signs until they reach the age of 6 months. And they almost never live past 5 years old. And this disorder causes the destruction of the central nervous system. One of the first signs of this disorder is a red spot in the back of the baby's eye, and you can actually see this. The disease is on chromosome 15 and is caused by the absence of hexosaminidase A, also known as Hex-A, which I'll be referring to. And this is an enzyme that breaks down acidic fatty acid materials called gangliosides. And a normal healthy child, these acidic fatty acid materials, gangliosides, are created and then broken down by the enzyme Hex-A. So these gangliosides are created and then broken down by the enzyme Hex-A. And this is happening while the brain is developing. So that's an important key, that this process of building up these fatty acid materials and then breaking them down is happening while the brain is developing. Now, that's how a normal child would develop, and that the fatty acid is broken down as the brain is developing. But a child affected by Tay-Sachs, this enzyme Hex-A isn't working properly. Therefore, there's a buildup of gangliosides, and this causes the central nervous system to be affected. Physical and mental abilities then decline, because if you're affecting the brain, you really are affecting both. And the development of the child will slow once symptoms begin. So again, it doesn't really show signs until 6 months years old, and then very rarely do they live past 5 years old. And so the development of the child will slow once the symptoms begin, and then it will stop and even reverse, that their development will, it doesn't improve, it really regresses. And there's no cure for this disease, and it's considered extremely rare and is known to affect people of Jewish heritage more than any other population. So definitely a very, very sad one, because this one is affecting kids. And even though it is rare, it is known. It's one of the more well-known genetic disorders. But that's for your information. Well, that wraps up today's episode of DNA Today. You can tune in next week Monday, and every Monday at 1 p.m. on whus.org or 91.7 FM to learn more about genetics. You can check out the website at dnapodcast.com. It's full of content, including all episodes broadcasted, as well as mini lessons I've done previously on there, and that's all about genetics. And there's resources to expand your genetic knowledge. Start here and expand from that. You can follow on Twitter at DNA Podcast. You can also follow me on Twitter at Kiradinen, Kiradinen. If you have any questions you want answered, I love getting emails. It is very exciting. I may or may not jump up and down in my dorm room when I receive them. Love hearing from you guys. So if you have questions you want answered on air, or you want to comment on something, or you found something really interesting, you want me to learn more about it for you that I can share on air, or just send you a couple links to be like, this is what I found in my research for you. You can shoot me an email at info@dnapodcast.com. Again, those resources are really great. The website is where I want to point you to the most, because all this information is on the website. So you can just poke around on there, and I have all of the episodes archived. So you can, anything that you've listened to on air here, you can go back and listen to again if you didn't quite understand any news story, and sometimes it's good to re-listen to it. I definitely do that with a lot of YouTube videos when I'm studying, that you watch it once and you're like, all right, I want to do that again. Especially when I do lessons. I went over the genetic disorder really quick, but for other kind of lessons that it's good to re-listen to, because sometimes I go a little bit fast. I feel like I've been talking very fast on this episode, but I'm excited, so sometimes it happens. So yeah, definitely check out dnapodcast.com. And thank you for listening. You can join me next week, Monday at 1 p.m. to learn and discover new advances in the world of genetics.